This is a single HIV virus. It has a nuclear envelope that surrounds the whole cell, and under it is a layer of proteins. In the center is a bullet-shaped protein that protects the two strands of RNA and an enzyme that is called reverse transcriptase and an enzyme called integrase. The HIV cell, when it enters the body, affects the body's T helper cells. It does this by taking the receptors on the outer envelope to connect it to the cell's CD4 receptors. This is what allows the viruses to connect to each other. The second receptor, called CCR5 in blue, then connects to the virus's receptors. Then the receptors pierce through the membrane, allowing them to connect and pull the two closer together. When the two membranes fuse, the insides of the virus are then injected into the cell while the envelope protein is left at the surface of the cell. When the insides of the virus enter the cell and the two layers of the proteins are dissolved inside the cell, the enzymes and RNA are then released into the cell. The enzyme reverse transcriptase takes hold of the RNA and converts the RNA into a single strand of DNA. As this happened, random errors occur because of its sloppy decoding. That same RNA then goes through the same process again to make a double strand of DNA. After this occurs, the other enzymes that came with the virus, called integrase, takes hold of the DNA and brings it into the nucleus of the cell through a pore. Once in the cell, integrase takes the DNA and randomly puts it into the cell's DNA. That is what ensures a lifelong infection. Then when the strand of DNA is transformed into mRNA through a protein, it is sent to a ribosome. That RNA is then coded to make an envelope protein that is brought to the surface, and it starts to make the beginnings of the HIV cell. As this happens, though, other messenger RNA are being made and are taken to the ribosomes. These RNA decode to make viral proteins that make up the rest of the HIV virus. Once these proteins are made, they are transferred to the surface of the cell where the envelope proteins are located. Some of them even take some little strands of RNA to make the viruses. That virus is then branches off and to make its own HIV virus.